In this exciting video module, we'll be talking about fertilization. So here we can see finally the sperm, after their long and arduous journey, have found the egg. What we notice first is that the egg is trapped inside of a protective membrane. This blue ring is a tissue matrix called the zona pellucida. Now this is really important in terms of immunology, actually. Remember that after meiosis, the egg has been transformed into something that is genetically unique. That's the whole point of meiosis, to produce genetically unique gametes. Now, inherent within that process, the egg is vulnerable to attack by the maternal immune system, right? The mom's macrophages within the fallopian tube or uterus would immediately attack the egg, recognizing it as a foreign invader. So nature is really smart. It has a shell around the egg. Surrounding the shell is the corona radiata, the radiating crown. This is actually a collection of hundreds of cells called the granulosa cells. And if you've watched the BBC fertilization video, you'll recognize them as sort of like water balloons, sort of orange and yellowish, squishy, fluid-filled cells. That's what we see here. In this illustration, they're shown with a slightly purple nucleus and this tannish color. Okay, so we've got the egg, the zona, and then the corona radiata. The sperm have to get through those outer layers if they have a chance of injecting their nucleus into the egg. This is really what fertilization is. Once that package is delivered, the egg is said to be fertilized. There are actually a couple of steps after that. Remember the meiosis II of the egg, and then the sperm's chromosomes in the egg's chromosomes will come together. Here we go. The first sperm to arrive on the scene cracks open his acrosome, beginning the acrosomal reaction so that he can chew through the corona radiata. And that pink foamy reaction will continue. Hydrolytic enzymes digest through the zona pellucida. The cortical reaction is the next thing that's going to happen. Once that first sperm cracks through the zona pellucida and sort of slithers on to the surface of the egg, again, I refer you to the BBC fertilization video. They do a great job of showing how that happens. The egg has prepared all of these little landmines. It's sort of silly, but I imagine that these are lime jello filled landmines. Okay, these are the cortical granules, and as soon as the first sperm touches this membrane, they all start bursting, injecting their sticky lime jello into the space between the plasma membrane of the egg and the zona pellucida. That will fill up the space, preventing any other sperm from touching the egg, and the cortical reaction also hardens the zona pellucida, so any other sperm who were on their way through are turned back, and it is by those two processes that the egg guarantees that she'll only get fertilized by one sperm. Now this is really amazing if you think about it. The egg has designed mechanisms to prevent too much DNA from getting inside. We could imagine if a situation where two sperms simultaneously got in, or within a few seconds of one another, both of those sperm pronuclei would end up inside of the egg, and there'd be way too much genetic material, right? The female chromosomes wouldn't know who to line up with, and that wouldn't develop into a normal baby. It probably wouldn't develop at all. So nature's smart. It knows that there is the potential for many sperm to try to be hacking in, and so this cortical reaction delays those second and third sperm on the scene from actually fertilizing the egg, thereby guaranteeing that the egg is only fertilized once. Well, then what happens? If we flip forward to this graphic, we can see the next 6 days or so of the very first stages of embryological development. So remember, just before fertilization, the egg was in this form, the ovulated form, the secondary oocyte. And you might now recognize that the zona pellucida was present from even before ovulation, but we can see it here. The first polar body, having been ejected from the cell, is still stuck inside of the zona. Here come the sperm, they're cracking in, fertilization 0 hours when the very first sperm injects his nucleus and then his DNA. We can see here the egg then finishes meiosis 2, creating the second polar body. So at this point in the scene, it looks a bit strange. There's one cell with 4 nuclei inside. First polar body, dead and meaningless. Second polar body, lost the meiosis 2 battle. Her chromosomes are going to be garbage. Here's the winner, the egg's pronucleus, a haploid nucleus containing 23 single maternal chromosomes, and the sperm who won the race has brought his 23 single chromosomes onto the scene. That's all still happening within the zona pellucida. Once the maternal and paternal chromosomes match up for the very first time, we have restored the diploid status. There are now 23 pairs of chromosomes. And at this point, we can call it a zygote. It is a fertilized egg. Technically, it's not an embryo yet. Being very academic, we sort of change the name as we go through time. So it starts as a zygote, then it becomes a blastomere. You can see the first few mitotic divisions happen still within the zona. 72 hours later, we have a whole bunch of cells, all having divided up the cytoplasm from the original egg. These are very small cells, but they're getting ready for the very first time to begin to express their genes in different ways. We call this specialization, right? When a generic cell expresses genes to become a more specific form, and her twin sister expresses different genes to become something else. Differentiation is another way of talking about that. So at this point, these are all undifferentiated pluripotent stem cells, but they have to crack out of the zona and inflate themselves into this crazy structure called the blastocyst. The best way that I can think of describing this is akin to a beach ball. Imagine you order a beach ball from Amazon. It comes in a tiny box. You open the box and inflate the beach ball. Look, it's really the same structure. This is just collapsed, and this is just inflated. And in the metaphor, the box is the zona pellucida. Once that cracks out and inflates itself, the zona is its job is now done. But there is a short period of time when this exposed blastocyst, this new foreign creature, is potentially going to be scarfed by the maternal immune system, the uterine macrophages. So this has to happen pretty quickly. Once inflation into the blastocyst happens, it has to land on the surface of the endometrium and then sort of burrow down so it can hide a little bit within the epithelium. So this whole story is called the migration of the Conceptus. Conceptus meaning conception, the thing that was created in conception, this little zygote. Just food for thought. There's a lot of debate in the public square right now and in politics about when life begins. Some proposed personhood amendments would make it legally recognized that life begins here at the zygote stage. This is a unique genetic complement, and therefore this one cell should be granted all of the rights of the government, of our federal rights. They're called personhood amendments. Really interesting. I don't know the answer to it. When does life begin? When should we recognize this developing ball of cells as an individual? At conception or at implantation? A lot of interesting questions, and science can really maybe help us understand what's going on here. Okay, so just food for thought. Here's the next thing we want to talk about, implantation in a little bit more detail. So here's the developing blastocyst. At this point in time, we only have two different types of cells. These have so far only differentiated into a couple basic types of cells. Can you tell which one is going to develop into the baby? Of course, it's the embryoblast. The surrounding trophoblast, shown here in purple, is going to develop into the layers that surround the developing embryo and fetus, connective tissue membranes and epithelium that will tap into the mother's blood supply and ultimately become the placenta. So at about 6 to 7 days, the little eagle has landed, and now she begins to invade the mother's body, the very strange structure called the syncytiotrophoblast. Pay close attention to the color here. It will help you understand this process. Look at that green color. Going from this invasion of little roots, the syncytiotrophoblast, we now see that has completely surrounded the developing embryo. On the inside, we see some tissues that are developing into the placenta and some of the vascular structures. And for the first time, we can see the tricolored disc right here in the middle. This is what will actually develop into the baby's body. It doesn't look like much right now. The blue sac above will develop into the bag of waters, the amniotic sac. And the yellow bubble below will develop into the yolk sac. Look at this, just like a chicken's egg, there is a yolk within any vertebrate embryo, right? A yolk, just like the chicken's egg, serves as a source of nutrition for the developing embryo. I don't know if you've ever thought about it before, but you know that mammals, including human babies, are attached to the mother during embryologic development with an umbilical cord. But what about chicks? After the mom lays the egg, how does she pass nutrition to the baby? Well, she doesn't. All of the nutrition has to be there from the beginning, and that's what the yolk is. So as a chick develops, it absorbs the yolk through its abdomen, through its functional umbilical cord. And all of that nutrition is transformed into the little chick who hatches out. At that point, there's no yolk. Okay, so in human embryology, it's actually quite similar. The yolk will develop into a larger structure, like we can see right on this slide here. Look at the yolk sac. It's attached to the developing abdomen of the baby. This will be the baby's head. This will be the baby's lower pelvis and sacrum. And anyway, the yolk sac, like the chick, is absorbed into the body of the developing baby. And we can see it here at this stage. Just a few weeks in, the placenta has already developed. The bag of waters has now completely surrounded this thing that looks a lot more like a baby now, even though it's only about the size of a jelly bean. And the yolk sac, having served its purpose for the first few weeks of development, is now being completely absorbed. It will atrophy and disappear. And then throughout the second and third trimesters, we're going to see, and even the latter part of the first trimester, we're going to see that the placenta then serves as the primary source of nutrient and gaseous exchange with the baby. Mother's blood supply comes through the uterus, and although mother's blood and baby's blood don't mix, they come very close to one another through this interface, which is really what the placenta is, an interface exchanging oxygen for carbon dioxide, glucose and amino acids for wastes like urea, and then through the umbilical cord and tied directly into the baby's circulation. These crazy vessels, the umbilical cord and the umbilical vein. So that's sort of a crazy overview of fertilization and the basics of embryology. In the next video, we'll take a closer look at some of the details of human embryological development.